So welcome back to Unit 2, Cell Structure and Function. This is the second video in the series, and today we're going to be talking about cell size, plasma membrane, and membrane permeability, which is subunits 2.3, 2.4, and 2.5 of the AP Biology curriculum. All right, so to get started on talking about cell size, let's make sure you know how to calculate the surface area and volume of various shapes. These formulas will be provided to you on your formula sheet, so you don't need to memorize these, but you do need to use them for calculations. So let's practice by calculating the surface area and volume of cubes of various sizes. So here I have drawn, and it's not perfectly to scale, but cubes that have a size or a side of 1 centimeter, 3 centimeters, 6 centimeters, and 10 centimeters. So for each of these cubes, what is the surface area and what is the volume? So let's start by just going over that formula. So the formula for calculating surface area of a cube is side times side, which gives you the surface area of one side, times 6, because there are 6 surfaces of a cube. To figure out volume, it's length times width times height, but because in a cube those are all the same, it's just side times side times side. So for a 1 centimeter cube, the surface area is 6 square centimeters, and the volume is 1 cubic centimeter. It's very important to have those units correct because it's showing for the volume, it's a cubic measure. There are 3 dimensions of it. Whereas for surface area, there are just 2 dimensions. That's a flat area. It's on 6 different sides, but it's still just a flat area. You could sort of unroll it and it would just be flat. There's no 3rd dimension there. All right, so the values are also shown for 3 centimeter and 6 centimeter and 10 centimeter. My video, it looks like, is kind of covering up the volume of the 10 centimeter, but that's 1,000 cubic centimeters. You just get that by 10 times 10 times 10. All right, so let's go on and try to apply this to a situation, to a lab. All right, so in this lab, what you've done is you've taken some agar cubes of various sizes, and they were pink to start with because they had an indicator in them, a pH indicator, and then you put them in vinegar. And the vinegar is acidic, and so it turns that indicator from pink to white. And so as the vinegar diffuses in, the agar cube gradually turns from the outside in from pink to white. And you can see these cubes here spent some time in the vinegar, but they didn't spend long enough for the vinegar to get all the way to the center of any of these cubes. All right, so let's look at some actual numbers. As the size of a cube increases, what happens to its surface area and what happens to its volume and why? So try to graph each of these. How does the length of one side affect the surface area of a cube, and how does the length of one side affect the volume of a cube? So here we see that for each of these relationships, we have direct relationships. As one gets bigger, the other gets bigger. As a side gets bigger, the overall surface area gets bigger. And as the side gets bigger, the volume gets bigger. This makes sense. These are just bigger cubes. A smaller cube is going to have less surface area on it, and it's also going to have less volume. The bigger the cube, the bigger the surface area, and the bigger the volume. So let's make this one step trickier, and now let's look at the relationship between the length of the side and the ratio of surface area to volume. Try sketching out this graph. So here we see we have an inverse relationship. As the length of one side increases, the surface area to volume ratio decreases, so that we have this sort of downward sloping graph. If we look at each of the cubes, we can sort of figure out why. If we look at our 1 centimeter cube, our surface area is 6 centimeters squared, and our volume is 1 cubic centimeter. So 6 centimeters squared divided by 1 cubic centimeter is 6. Whereas if we look at our 10 centimeter size cube, we have 600 over 1,000. That's only 0.6. So the larger the cube, the smaller the surface area to volume ratio. So let's do some sort of a summary. Why is this, first of all? Why is it that those first 2 graphs show direct relationships, but the 3rd graph shows an inverse relationship? And then also, kind of why does this matter? How does this relate to the understanding that cells are small? So to do that, to explain how cells are small, relate it to that agar cube lab. See if you can apply that agar cube lab to actual cells. So in our agar cube lab, we found that even though the rate of diffusion is the same, the vinegar is moving from the outside of the agar in towards the center at the same speed, right? The same number of centimeters per minute. If it's a smaller cube, the center is kind of not as far away from the outside. And so that surface area to volume ratio being larger for a small cube means that in the same amount of time, we're going to diffuse into a bigger percentage of that cell, or a bigger percentage of that agar cube. For large cells, even though it's still diffusing at the same rate, the middle is relatively so far away from those edges that in the same amount of time, you're going to diffuse into much less of the cell. So this is a concern. If cells are too big, it's going to take too long for materials to get all the way to the center. So that's not going to have an efficient exchange of nutrients and waste products. So that's really why cells are small. All right, we can also see that here. We can see the diagram at the bottom showing that a smaller cube has a shorter diffusion distance. The center is relatively closer to the outside. But we also have a couple of other sort of examples of the application of surface area to volume ratio. So what are some advantages and disadvantages of high surface area to volume ratio? I would like you to take a look at the plants. So compare the shape of the plants and propose an ultimate explanation, relate surface area to volume ratio. And then also look at the comparison of the position in which these dogs are sleeping. How does that relate to surface area to volume ratio? So for our plants, you'll notice that the plant on the left has a very large surface area to volume ratio. Almost all of the plant is kind of on the surface because it's so flat. Right? That means it's great at catching sun, but it also means that there's a lot of evaporation. That's going to be a problem for desert plants. So if you look at the cactus on the right, that's sort of much more of a sphere shape, that's going to hold on to liquid better because it has a lower surface area to volume ratio. So flat surfaces have a high surface area to volume ratio, and spheres have a comparatively low surface area to volume ratio. So it can hang on to that water. Similarly, we can look at the position of these dogs. So the dog on the left is kind of laying flat. So much more of its body is touching the environment, both the floor as well as the air. Whereas the dog that's all curled up is more sphere-like. So it's exposing less of its surface area to the environment. So what might be happening for the dog on the left is that maybe it's a hot day and the dog wants to have a lot of evaporation to help cool down. Maybe that floor is a little bit cooler, so if it exposes more of itself, it can regulate its temperature better. And maybe the dog on the right, maybe it's cold out, and so it wants to retain its heat. It doesn't want to lose heat to the environment. It wants to save that. So it's in more of kind of a sphere shape where it can hold on to more of its heat because it has a lower surface area to volume ratio. So let's continue our discussion of how structure informs function by looking at 3 more examples and focusing on how different organisms have evolved to obtain nutrients and eliminate wastes. So let's look at root hairs, guard cells, and small intestine villi and microvilli, and see if you can, from these pictures, try to figure out what's going on and relate structure to function in one of these examples. In root hairs and in the small intestine, we can see that there are kind of protrusions. So there are areas kind of folded or sticking out into the environment. In the case of root hairs, it's that there are these little root hairs sticking out into the soil from the plant. And in the small intestine, there are these folds of the small intestine called villi, and then even smaller little protrusions called microvilli on the surface of the villi that protrude into the lumen of the small intestine where the food is. So in both of these cases, this is a way to increase surface area to volume ratio by having these folds or protrusions to obtain nutrients from the environment. If we look at guard cells, this is slightly different. What we have here is a way of plants controlling how exposed they are to their environment. So if we have stoma opening up, that's going to allow the passage of gases in and out of the plant. If they're closed, then we can't get gases in and out of the plant. And so then the plant, based on the conditions, both internally and externally, can balance how much exchange it wants to have with the environment, right? So it might want to take in some carbon dioxide for photosynthesis, release oxygen, and keep that balance of gases, but it also has to balance how much water it's going to lose. And so the way that these are controlled is by the size of the vacuole in those cells. And so if the cell has a large central vacuole, it kind of puffs up the guard cell to make it more rigid, to make that opening there. So it can control the shape of that cell through the vacuole, and that controls sort of the function of that gas exchange. So let's move on to section 2.4, which is the plasma membrane. Let's start with a review. Look at figure 1. What do parts 1, 2, and 3 represent? And then how does that relate to figure 2? So figure 1 is showing a phospholipid. The purple part on the left, part 1, is showing that phosphate head. You can see the phosphorus there. There's also a head group that includes nitrogen, some carbons. We're not going to worry much about that head group. Head groups can vary and can influence function, including signaling, cellular recognition, but we're not going to worry too much about that. All right, number 2, that sort of pink part, is glycerol. That's that connection in a lot of lipids. And what that's connected to is the phosphate on the left, and on the right we can see our 2 fatty acids. So this is a phospholipid. All right, how does this relate to figure 2? Well, all of the little red molecules with the 2 yellow tails are molecules that we see in figure 1. So these are all phospholipids. So part 1 of figure 1, that phosphate head, is the little red ball portion. And that's polar, so that's going to interact with water both on the inside and the outside of the membrane. The yellow tails, or the fatty acid tails, those are nonpolar. You can see all those carbons and hydrogens shown there. So those are going to be hydrophobic and hide from water. So those will be on the inside. What is the function of a membrane, and what other molecules are found in the plasma membrane? And then I also want you to talk about how does the structure of the plasma membrane relate to its function? And finally, what are integral, peripheral, and transmembrane proteins? Some of these questions you may be familiar with already, others may be new to you, but just give it a shot. So answer those 4 last bullet points there. All right, so at the basic, a membrane separates 2 different sides of a cell or an organelle, right? So the inside and the outside, it gives a barrier so that we can have a different environment on one side of the plasma membrane than the other. What other molecules are found in the plasma membrane? Well, we can take a look at the diagram for that. We can see that there are quite a few proteins. That's going to be important in all of biology to understand what kind of proteins are in the membrane, and we'll go into more detail on that in a little bit. You can also see that there's cholesterol in there, and cholesterol helps kind of keep the membrane fluid. It helps it from getting too solid. How is the structure of the plasma membrane related to its function? Remember the fluid mosaic model? So all of these pieces can move around. So the plasma membrane is very flexible. You can almost think about it like a fairly sturdy bubble. So it can change shape, it can get bigger and smaller if we have more or fewer molecules. The components within it can move around, so there's a lot of flexibility here. All right, and finally, what are integral, peripheral, and transmembrane proteins? You might be able to figure these out just from those words, right? If you know what integral or integrated is, if you know what periphery is, and then the word trans, the root word trans, means across, and membrane, we know, is our membrane. So let's take a look at those. All right, so here we have a picture in more detail of some of the membrane proteins. So let's look at the peripheral versus the integral membrane protein. So an integral membrane protein is permanently embedded within the lipid layer. You can think of it as integral. It's required in there. It stays in there. Whereas peripheral is on the outside. It's temporarily bound to the lipid bilayer, or it's temporarily bound to one of those integral proteins. A transmembrane protein, if we look at the root there, again, trans means across, and so a transmembrane protein completely spans the membrane. Here we have another review term. What is a protein domain? So a protein domain is a region of the protein. So we can see that in this diagram, that there's, for example, on the left, it's showing a hydrophobic region. And so the hydrophobic region of that transmembrane protein is going to stay on the inside, right, with the fatty acid layer. Whereas the hydrophilic region is going to be on the outside. And so if you have a multi-pass protein that goes in and out of the membrane and connects on both sides, you will have a series of hydrophilic and hydrophobic regions. And some of these are well conserved, like you'll see that same exact sequence of amino acids, that same region in multiple different kinds of proteins. All right, so let's go on and look at the wonderful variety of membrane proteins. So what are different things that membrane proteins can do? Take a look at these diagrams, and we're just using this as diagram interpretation practice, so I'm less concerned about specific terms and more about take a look at the pictures and see if you can figure anything out about them. And as a note, our primary focus is going to be on that first one, on A for right now. So what we see in A is transport. We see that because we have molecules moving from one side of the membrane to the other. There are different kinds of transport, and we'll get to those details later on in this unit. In B, we see that the proteins are sort of causing changes in molecules. They fit into specific spots, something happens, and the molecule changes. So it starts as a triangle, comes out as a square, then goes into another part of the enzyme, it comes out as a circle. So this is causing change. All right, we can also have signaling. We're going to have a whole unit later on in the year talking about signal transduction and cell signaling. So that's an important role of membranes or proteins in the membrane. We also have recognition. So proteins serve to recognize one another, and that's cell-to-cell recognition. So you can see that in part D. You can also have proteins connecting two different cells together. So intercellular, remember inter is between, so connecting two different cells together. And finally, we can have proteins that attach various aspects of the cell all together to kind of keep that cell sturdy and keep that cell, all of the pieces together. So it can attach to the cytoskeleton and the extracellular matrix within the cell or outside of the cell. All right, so now we're moving on to our last unit of the video, which is section 2.5, and we're talking about permeability. So selective permeability, think about those words and think about how this relates. How do molecules get from one side of a membrane to the other? And what factors influence that movement? So there's a lot going on in this slide, but the major takeaway is, that, or at least that I want you to get right now, is that the structure of the molecule impacts how it gets from one side of the membrane to the other. Specifically, if you have a nonpolar molecule, that can go straight through the membrane. So if you look at the left side, part A, we have simple diffusion of fat-soluble molecules directly through the phospholipid bilayer. So for example, steroids that are lipids can go right through that membrane, that they don't need to have protein channels. This is also true of small nonpolar molecules like carbon dioxide and oxygen that can go right through that membrane. Other molecules, such as sugars, amino acids, ions, water, these are all going to be polar and lipid insoluble. So these are the polar molecules are hydrophilic and associate with water or are water themselves, and they're going to need proteins to help them through, that they can't pass through on their own. And so we have different methods here. One is kind of a carrier that changes shape to allow a specific molecule through. Others are kind of channels, so they have a specific size and also specific polarity within that channel to allow specific molecules through. Water is a special case. Water is so small, it can technically sneak through that membrane even though it's polar. However, it does so slowly that most of the water passing in and out of a membrane is through channels, is through protein channels. And that channel has a specific name called an aquaporin. So aqua meaning water, pore meaning hole. So an aquaporin is a channel specifically made for water. And the movement of water is called osmosis. So we'll talk about diffusion later on in the unit. And specifically, the diffusion of water is called osmosis. So the final slide of this unit is talking about the cell wall. So we've talked a lot about cell membranes, but I also want to make sure we talk about another sort of coating on the outside of a cell, which is a cell wall. So first question, do all cells have cell membranes, and do all cells have cell walls? After that, what's the function of a cell wall? And finally, and you can get this from this picture, what macromolecule are cell walls made of? So it's important to know that all cells have cell membranes. That's a defining characteristic. Cells have cell membranes, all cells. Some cells have walls and some don't. So a lot of non-animal cells have cell walls, and they help provide additional support and additional permeability barrier to control what molecules come and go. The walls of plants and fungi and prokaryotes are composed of carbohydrates. So we can see in this diagram a long string of those glucose rings. Remember, the natural form of glucose is in that ring form. And if you have a whole bunch of those bound together, you get a polymer. A polymer of carbohydrate is a polysaccharide. Now, there are different kinds of polysaccharides, and the type of polysaccharide that is sort of the most common composition of cell walls is called cellulose. So vertebrates do not have the enzymes necessary to break down cellulose into its glucose components. However, some bacteria do. And so organisms that eat a lot of plant material and live off of plant material, let's say like cows that eat a lot of grass, they can't break down that cellulose themselves, but they have gut bacteria that can. And so the gut bacteria is breaking down that cellulose, and then the cows are able to extract the energy from those monomers, from the glucose monomers. So that's going to be it for today. As a reminder, this video covered sections 2.3, 2.4, and 2.5 of the AP Biology course, which included cell size and the plasma membrane.